Hello, my name is Emily Wisz, and I am going to be talking to you a little bit about age differences in electrophysiological correlates of visuomotor adaptation. So starting off, motor learning is a specific type of learning that occurs through the repetition of movement. Learning of motor skills is accomplished through two phases, a fast process and a slow process. The fast process occurs first and allows performance improvements to occur rapidly by adapting strongly to errors. This process, however, does not help to maintain memory of the movement over time. The slow process responds to errors more gradually and provides slower gains in performance over time. The slow process may contribute to solidification of motor memory. This implies that different parts of the brain are responsible for initial motor learning and later motor recall. While the neural mechanisms associated with early memory learning are not fully understood, previous research in our lab suggests that there are changes in the neurophysiological signature of performance across learning and healthy aging. To examine how these neural mechanisms change across the lifespan, we compare motor learning performance and neural activity between healthy older adults and healthy younger adults. EEG is a non-invasive method of examining neural activity, so EEG recordings provide us with event-related potentials, or ERPs as they might be referred later on. Event-related potentials show us the time course of changes in neural activity related to changes in cognitive function. For this study, we were most interested in an event-related potential called P300, which has an associated and attention control mechanisms and processing of task context information. Previous research has shown that a posterior P300 component called the P3B is reduced as a motor learning task progressed, which is indicative of improved learning. Our hypotheses were that older adults' initial adaptation to visuomotor rotation would be slower than younger adults, and secondly, that learning-related modulation of the P3 component would be more pronounced in younger. Moving on to the methods in this study, participants completed a visuomotor rotation task on the kin arm robot, which can be seen in the method section. While using the kin arm, participants grasp a handle attached to robotic motors, while visual stimuli are displayed on a horizontally oriented screen located just above the handle, matching the plane in which reaching movements are made. Participants were asked to use the handle to move a cursor from a starting location to a target location. The first task includes some learning trials where the cursor moves congruently with the participant's hand, and then in subsequent trials, however, the cursor was rotated by 45 degrees in a clockwise or counterclockwise direction from the participant's movement of the handle. The goal of this task was to move the cursor straight to the target location. That means that the participants have to learn to aim 45 degrees in the opposite direction over time. Gradually, while participants were performing this task on the kin arm, continuous EEG recordings were required using an electrode EEG system. So moving on to the behavioral data, I would now like to direct your attention to the figures in the middle of the poster. The top figure shows how younger adult and older adult performance differed on the visuomotor rotation task. We are most concerned with this middle section, which shows how participants adapted to the cursor rotation. Overall, younger adults adapted to the rotation faster than older adults initially. The at-risk shown represent significant differences between the groups at both the early part of the adaptation and at the later part. The first controlled mainly by the fast system and the second controlled mainly by the slow system. These findings are consistent with previous research showing a reduction in early adaptation in older compared to younger adults and suggest that later adaptation may also be reduced. ERP data-wise, the figures shown below show the changes in ERPs during the adaptation phase of the motor learning task of the younger adults and the older adults on the right-hand side. The P300 was most pronounced for both groups over the posterior piezoelectrode site, consistent with previous literature suggests that visuomotor adaptation elicits the P3B component. This component was also modulated across adaptation, such as the amplitude was reduced as people learned the visuomotor rotation. We ran a statistical analysis of this P3B modulation by comparing younger and older adults across quartiles of epochs in the adaptation phase. Although the modulation of the P3B is more pronounced in younger adults, the effect was not significant, but this is likely due to the fact that there are preliminary data and sample sizes are small. Moving on to the discussion, consistent with prior research, older adults were slower to adapt to the visuomotor rotation initially compared to the younger adults. Also consistent with previous research, the P3B component was modulated across adaptation as participants learned. Our data suggests that this modulation is more pronounced in the younger compared to the older adults, although this effect was not significant in the small preliminary sample. Our preliminary findings suggest that the age-related changes in the learning-related modulation of the P3B component may help explain why older adults are slow to learn a visuomotor adaptation task. This interpretation is consistent with prior research that suggests that the P3B component is associated with the updating of working memory with task context information and learning. As we complete data collection in the future and increase the sample size in the younger and older adults groups, we expect that these observations will be more statistically significant.